My name is Bobbi Helgason. I'm a professor in the Department of Soil Science at the University of Saskatchewan, and I co-lead the Root Microbe-Soil Interactions Flagship together with Stephen Siciliano. We're all really excited to get started this afternoon, but first I'll just take a minute or two to go over some housekeeping items and to orient you to our virtual format. So the very first item of note is to just let you know that all of today's sessions are being recorded. The attendee microphones are muted during the sessions, but you are able to interact through the chat function, which I'll explain in a minute. So the portal where you access today's session will be your landing place where you can access links to all of the live sessions and the recordings after the symposium. If you're having any technical difficulties, you can click the question mark button on the web player, and that will redirect you to the help page. If you aren't using the media player and you're just on the login or the web portal, there's also a blue help button at the lower right-hand corner of your portal screen. While you're in the portal, please be sure to go and check out our poster gallery. Just like in past years, we've got a voting poll for fan favorite posters, so take a minute to vote, if you will. It's a great opportunity for us to acknowledge all of the very good work of our teams and to showcase the excellent breadth of work in the P2IRC program. I mentioned a minute ago that attendee microphones are off, but we really encourage your questions and interactions with the speaker by using the chat box feature in the media player to submit your questions and chat with other attendees. You can also click the Ask a Question button, looks like a hand, located in the media player to submit a question for the panelists. Please feel free to post your questions during the presentation as well as during the panel session. And slightly different from this morning's format, between each of the presentations, we're going to take about a 30-second transition period just to let you gather your thoughts and post any questions about the previous speaker. So again, that's just a 30-minute break, or 30-second break, pardon me. We'll put up a break slide, so don't step away, we'll be right back. We're all getting used to this new way of doing things, so please do post your questions. The conversation will be so much better with your engagement. So that's it for housekeeping. I think we're ready to get started with our first speaker, Dr. Maggie Wagner from the University of Kansas, and she's here to talk to us about her work studying the interaction of the microbiome with heterosis effects in maize. With that, I'll sign off and we'll move on to the good stuff. Thanks.
My name is Maggie Wagner. I'm an assistant professor in the Department of Ecology and Evolutionary Biology and an assistant scientist with the Kansas Biological Survey, both at the University of Kansas. I'll be talking today about the relationship between hybridization and the microbiome in maize. Before I get started, I want to acknowledge several important people who played a big role in the work I'm going to talk about today, especially my postdoctoral mentors Peter Ballant-Curdy and Jim Holland at NC State University, my current collaborator Manuel Kleiner, also at NC State, and at KU, graduate student Kayla Klaus has done a lot of the experiments that I'll be presenting today. I like to start out my talks by showing these images, which make it really easy to visualize how the plant's phenotype becomes the habitat that's available to potential microbial symbionts. And these photos make it really easy to imagine how heritable plant traits such as the stickiness or permeability of the leaf cuticle, stomatal behavior, and the topography of the leaf surface can all have big effects on the habitat encountered by these microbes. Heritable plant traits can also influence the microbial habitat available below ground. As soon as a plant starts growing, it begins to influence its immediate soil environment surrounding the roots, also known as the rhizosphere. It does this by secreting many carbon-rich compounds and other phytochemicals such as hormones, as well as altering the water and nutrient status in that soil zone. As a result, only a subset of the extremely diverse ambient soil community is able to colonize successfully the rhizosphere soil because it's a more specialized environment. And zooming in even further into the interior of the roots, an even smaller subset of this microbial community is able to colonize there, where other heritable plant traits such as the immune system come into play. This winnowing down process is partially influenced by stochastic mechanisms such as founder effects. However, the genetic variation within the plant host that determines the plant's phenotype also has a deterministic effect on microbiome formation. As a result, two plants growing side by side within the same ambient microbial community can end up forming different microbiomes if they differ for a functional trait that influences that microbial habitat. This framework of the reciprocal effects of plant genetic variation on microbiome composition and function forms the foundation of the work that I'm going to be talking about today. So first I'll talk about some results from a project looking at how hybridization affects the formation of the maize microbiome. And next I will talk about the reverse direction of causality. How does variation in the microbiome affect phenotypes? And for both of these I'll be focusing on the specific context of hybridization and heterosis. So how can hybridization affect the microbiome? This hypothesis assumes that at least some traits showing heterosis will also affect the composition or function of the microbiome. So in the absence of heterosis, the F1 hybrid will take a trait value intermediate to the two parents, and as a result will form a microbiome that is intermediate in composition to the distinct communities formed by its two inbred parents. In this cartoon, the differences in microbiome composition are illustrated by the abundance of this larger pink microbe that you can see in the host-associated communities at the bottom. And in the case of mid-parent heterosis, the hybrid will take a trait value similar to one parent or the other, deviating from the mid-parent expectation, and we hypothesize that this would result in a microbiome that is similar to that of one parent but not the other. And finally, with better parent heterosis, where the hybrid value exceeds that of both parents, you would expect the hybrid to have a microbiome that differs in composition and function from both parents as well. So within this framework, there were two major questions that I wanted to know the answer to. Firstly, are there consistent differences between the microbiomes of inbred lines versus maize hybrids? And secondly, looking within individual crosses, can we tell which microbiome features, if any, show evidence of heterosis? To answer these two questions, I set up a field experiment involving 18 different genotypes, 7 inbred lines, as well as select F1 hybrids between pairs of them using B73 and MO17 as the two common parents as shown here. So first I wanted to use this data set to ask this question: are there consistent differences in the membership of those communities between inbred lines as a group versus hybrids as a group? And so looking at the box plots on the right, these could easily be illustrating something like yield, but on the vertical axis we could also put any number of different microbiome features and test the hypothesis that these communities differ consistently in composition between these two groups. So on these two plots, each individual point summarizes microbiome composition for a particular sample, which came from either a hybrid or an inbred host, shown in green or purple respectively. And the leaf samples are shown in the left panel and rhizosphere in the right panel. This analysis showed that the bacterial communities in leaves overlapped almost perfectly between hybrid and inbred hosts. But in contrast, in the rhizosphere, there was a consistent shift in community membership between hybrid and inbred plants. To determine which organisms were driving these patterns, I fit negative binomial models to estimate the fold change in relative abundance of key taxa between hybrids and inbreds. I identified a few dozen taxa that were significantly more abundant either in hybrids or in inbreds. There were several interesting groups represented in this set. For example, Brady rhizobium has been known to fix nitrogen even in free-living contexts such as loose soil or rhizosphere soil. There were 3 different ASVs from the genus Burkholderia. It's known to include multiple plant growth promoting strains as well as multiple plant pathogens. So now, having established that there are stereotypical differences in microbiome composition between hybrid and inbred maize, we move on to the related but distinct question of which microbiome features show evidence of heterosis within individual crosses. To explain how I did this, it's helpful to revisit the framework I presented earlier, where functional traits showing heterosis translate into heterosis of the microbiome itself. We can actually test directly for evidence of heterosis in the microbiome using the same methods that we use to test for heterosis of the functional traits underlying the microbiome heterosis. So for example, we can use t-tests to test the hypothesis that a given microbiome feature of interest deviates from the mid-parent expectation that we have based on the microbiome composition of the two inbred parent lines. So this is what this test looks like for a microbiome study. Because these data sets are so immensely complex and high-dimensional, I started by reducing them to several summary statistics and testing those summary metrics individually. So for example, on this left panel, I've plotted the estimated marginal mean value of MDS1, which is the dominant axis from a principal coordinates analysis of community composition. So that you can think of this as a quantitative summary of community composition, which represents about 17% of the total variation in the data set. So I plotted this, or calculated this value for the female and male parents in purple and pink, as well as the F1 hybrid shown here in green. And you can see in this particular cross that the value for the F1 hybrid deviated from the mid-parent expectation. But looking at the other 10 crosses, you can see that this was not necessarily the case for all of them. There was variation among these hybrids for the degree of heterosis they displayed for this particular microbiome feature. Looking at a different summary metric of microbiome composition, saw a much more consistent result where all 11 crosses showed significant better parent heterosis for this particular component of the microbiome. So combining the results from all the summary metrics that I tested, a couple key patterns emerge. First of all, we see that heterosis for these microbiome features was more prevalent in the rhizosphere than in leaves, which is consistent with what we saw earlier when comparing hybrids versus inbreds as two groups. Furthermore, applying the same test to the relative abundance of individual ASVs showed a similar pattern, where heterosis was more common in the rhizosphere, and certain ASVs were much more likely than others to show heterosis in a larger number of hybrids. So to summarize this investigation of how hybridization affects the maize microbiome, we can say that host genotype explained up to 11% of the overall community composition. It's worth noting that while 11% is definitely low compared to the heritable plant traits that we're used to studying, that's actually a fairly high estimate of microbiome heritability compared to the previous work in other systems done to date. That wraps up the first part of this talk, where we were focusing on how genetic variation within the host plant affects the microbiome. Now we're looking at the opposite causality, where the microbiome in combination with the host genotype can influence the phenotype of the plant. And this project began when we were actually trying to figure out if we could recreate the results that I talked about previously from the field experiment using a highly controlled synthetic community, or SynCom, consisting of just 7 bacterial strains. So we did this in the lab in these gnotobiotic chambers where you can strictly control which microbes are able to enter the system. We added these 7 bacterial strains to the plants. We noticed something really unusual, which is that in the sterile control, which had no viable bacteria in the presence of the plant, there was no discernible difference in root system size between B73 by Mo17 hybrid versus the 2 inbred parents. And this is in contrast to the plants that did receive the live inoculum, where the normal pattern of heterosis was restored, where the hybrid had a larger root system than either of the 2 inbred parents. Connecting the dots, we can see that this effect was not because the synthetic community provided some growth benefit that the hybrids could uniquely take advantage of. Instead, what we see is that the synthetic community actually decreased plant performance, and this decrease was more pronounced for the inbreds than for the hybrid. To confirm that this was a real reduction in the strength of heterosis, we calculated the difference in mid-parent heterosis between the non-sterile and sterile treatments and conducted permutation tests under the null hypothesis that there was no real effect of the treatment. And what we found was that the reduction in heterosis that we saw was much more likely than would be expected just by chance. Next, we repeated the experiment using a different type of microbial inoculum. Instead of a highly artificial community of just 7 bacterial strains, we extracted a microbial slurry from a real farm soil and applied it to the plants in the same gnotobiotic chambers. As controls, we used an autoclaved aliquot of the exact same soil slurry, as well as a sterile buffer control. This experiment gave us a very similar result, where there was no difference in root biomass between the inbreds and the hybrid for either the sterile buffer control or the autoclaved soil slurry treatments. However, we did see the normal pattern of heterosis for the live soil slurry. And again, this was due to a reduction in root system size in the presence of live microbes. And this reduction in growth was more pronounced for the inbreds than for the hybrids. In addition to the effect on root biomass, we saw a very strong effect on germination success. Here I'm plotting the proportion of seeds that successfully germinated in each of the 3 treatments. The hybrid is shown in purple and the 2 inbred parents in red and blue on the sides. You can see that in the presence of the live soil slurry, there was an extremely low germination for Mo17, and germination success for B73 was about half that of the hybrid. But for the killed soil slurry, there was no significant difference between any of the genotypes. And in the sterile buffer control, there was only a significant difference between Mo17 and the hybrid. However, the magnitude of that difference was greatly diminished. Finally, we took this into the field to see whether we would see the same pattern in more realistic conditions. With the help of our collaborator Mark Hoffman at NC State, we applied 4 different fumigation methods to blocks of soil at the research farm in Clayton, North Carolina. Once again, we observed that reduction of the soil microbial community decreased the strength of heterosis for root biomass compared to the untreated control soil. And once again, the mechanism appeared to be due to microbes having a negative effect on root growth in the inbreds, but not so much in the hybrid. And interestingly, all 4 fumigation treatments also decreased mid-parent heterosis of both leaf number and plant height. So to wrap everything up, the work I've presented here demonstrates how the microbiome can act simultaneously as a host trait or part of the extended phenotype and as part of the host's environment. And as part of the host's environment, it can interact with host genotype to determine the phenotype that is ultimately expressed by the plant. This is certainly true in the specific context of hybridization, and the evidence suggests that the microbiome might be surprisingly relevant to our understanding of heterosis in maize. With that, I'm happy to take any questions. Thank you very much.
Good afternoon, and thanks for attending the PERC 2020 Symposium. Today I'll be talking about canola root bacteria communities and how they've been linked to yield performance. But because it takes a village, I'd first like to thank my PERC co-authors and the support sources that made this work possible. So understanding how plant root microbe interactions influence crop performance across trials is crucial in addressing threats to global canola production in the face of climate and environmental change. So here we used a novel analytical pipeline to assess bacterial communities during leaf development, flowering, and seed development, and the evolutionary characteristics that underlie these communities. And we tested several hypotheses. The first one, that Brassica napus selects several yield-promoting communities during development, that yield promotion is a convergent trait, and what convergent means is that ecological function has evolved independently of the phylogenetics. And 3, that host selective pressures shape the community assembly through time. So we sampled 16 genotypes across 4 trials, 1 in 2016, another 3 in 2017, and then in the lab we extracted 16S rRNA from the roots, and we spiked with Allia vibrio fisheri as an internal standard to allow us to compare abundances across samples. And then we did our bioinformatics using CHIME2. So out of CHIME 2, we get our species abundance table. We standardize relative to A. fisheri. And then we filtered low quality sequence data out to maximize our inferential power. And I don't have time to go through this, but the bullet point is that we reduced our number of taxa from 13,000 to just under 1,000. And then before we got into our modeling, we had to replace zeros using Bayesian approaches. Then we calculated our measure of yield performance. So this incorporates both yield stability and yield. And you want to be here, high yielding and highly stable genotypes. And we tested 3 different versions of this performance metric where we weighted yield preferentially. So we weighted the x-axis here, where we weighted stability, the y, and then where it was unweighted, where the best spot is really in this green square. And the spoiler alert is that yield-weighted performance was most highly associated with bacterial composition, so our downstream analyses focused on that. Then we wanted to determine which bacteria in each of these 3 developmental stages were associated with yield performance. So this is leaf development, flowering, and seed development. And then we get into our modeling and variable selection, where we modeled performance as a function of bacteria. And what we found was that there was a unique community in each of these developmental stages that was highly correlated with yield performance. But if you look to that same community outside of these stages, looking at the root mean square error, you find that that relationship disappeared. So there were 3 communities, highly influential, ostensibly of yield, but in different ways. And then finally, we looked at the phylogenetic characteristics of each subset community. So we quantified the demographic niche at each developmental stage and found that there were strong environmental pressures. There's a lot of separation between the environments in these 3 different stages. There was a total of 63 taxa across the 3 communities that were linked with yield performance, though no more than 27 at a given stage. And there was only 2, as shown by these gray rectangles here, that were shared between any 2 communities. And so despite the strong pressures and the taxonomic dissimilarities, when we look at the phylogeny, and I won't go into this, but essentially if your beta NTI values are high, that means that you've got clustering, so a very deterministic signal in your phylogeny. Below -2 is a very even community, so a lot of unrelatedness, and in between is largely random. So this suggests that despite the strong environmental pressures, the community assembly was largely dictated by stochastic processes. So the bullet point from this work is that we discovered what is likely a functional redundancy in the roles of bacterial community, in that there are multiple communities at different times that perform the same function, but in what seems like a different way. So if we revisit our hypotheses, we found evidence to support that Brassica napus selects not one, but several yield-promoting communities during development. We also found evidence to support that yield promotion is a convergent trait, so it's independent of the phylogeny. But we refuted this one, that host-selective pressures shape assembly through time. And so this does not follow the niche-based expectation that strong environmental pressure should result in a strong deterministic signal in the bacteria data. So this is unexpected, but very interesting, and could possibly pave the way to new breeding strategies for enhancing yield performance. So the next step already underway is to look more deeply into how these bacterial communities may be influencing yield using various omics approaches, and move us beyond the what are these communities doing and into the how are they doing it stage. Thank you.
Hello, I'm Kevin Stanley, professor in computer science at the University of Saskatchewan. I'm here to talk about inferring soil microbial networks, which is part of P2IRC Flagship 2, otherwise known as Flagship Unicorn. It turns out that soil is not just dirt, it's full of microbes. These microbes interact with each other and the environment around them. When they interact with crops, they can inhibit or promote crop growth. Understanding how these microbes interact with each other then is an important part of understanding how they could help or inhibit crop and food security. Well, how can we do that? Well, if you're on the savannah, inferring an ecological network would be easy. For example, you could observe a lion eating antelope and observe nothing eating elephants, and directly from those observations, build an ecological graph. However, in soil science, we're not so lucky. Soil scientists will take a soil sample, more or less throw it in a blender, and sequence the DNA. This DNA will tell you whether or not certain species are present. Unfortunately, we don't even really get species. We get amplicon sequence variants, or ASVs, which basically tell you whether or not a piece of DNA is different. From this, we get these very large tables, which have hundreds or thousands of taxa across the top, and usually dozens or hundreds of measurements along the horizontal rows. There's also a great number of zeros in these data sets, which makes this a very difficult problem. So how does one get from that table to a graph? Well, the simplest thing to do would be to run some correlations. This has been done. Unfortunately, correlations can lead to spurious edges in the graph. For example, you could end up with positive edges between a prey species with a predator species that eats the prey species' primary predator. To address this issue, Zhao et al. Proposed an entirely new mathematical framework for inferring ecological networks. They realized that in essence, we were actually looking at how different microbes change with other microbes through time and space. And in fact, this can be approximated by the Jacobian matrix. And we don't even need to know the full Jacobian matrix, we just need to know the sign at each element of the Jacobian matrix. They further observed that we could take pairwise differences from these taxas, row-wise, to generate deltas for each of the taxa, which would then be evidence for or against a particular sign pattern for a particular taxa. So how do we generate a sign pattern to test? Well, the simplest one would be to find all the sign patterns. Unfortunately, this scales as n through to the n, which should be chilling if you're a computer scientist, where n is the number of taxa. If you're not a computer scientist, here's a simple example. For a relatively modest ecosystem of 30 species, there are over 6 quadrillion possible sign patterns. If we could solve a sign pattern every microsecond, it would take 176 years to check them all. To solve this problem, computer scientists often use heuristics to search portions of the space which are more promising first. The authors of Zhao et al. Used a heuristic to infer sign patterns directly from the data. However, this only works if the data is nice-free. If you're a bioinformatician familiar with metagenomics, this assumption should be chilling. So how can we infer sign patterns better? Enter Umair Aziz, my student, who did all the work. Umair was able to first improve the efficiency of the algorithms and parallelize them so we could do more faster. He was then able to create better heuristics to infer better sign patterns faster, and was able to come up with a blockwise approach, which allowed a greedy solution to the problem by assuming that knowing that gazelles are eaten by lions does not tell you whether or not gazelles are eaten by leopards. From this, we were able to successfully create ecological graphs for microbial ecologies in PERC experiments. However, we found another problem with this particular approach, and that is that for many practical systems, the number of perfect solutions according to the quality metric is much greater than is useful. In essence, we've gone from looking for a needle somewhere in Saskatchewan to looking for a needle in a haystack outside of North Battleford. However, in computer science, we're used to looking for needles in haystacks, and we're confident that we will be able to solve this problem in the future. I'd like to thank all of my collaborators and you for your attention.
Okay, I'm Dean Chapman. I'll be telling you today about some of the technology development we've been doing around imaging at the Canadian light source and deploying something we call a phase-preserving beam expander, primarily to image the root system of plants. So if you're familiar with the synchrotron or not, the beam that comes out of the synchrotron is quite small vertically, and the type of sources that we have at the biomedical beam line provide a wide beam but vertically small. And so that's very handy, but what happens if the thing you're imaging doesn't fit in the vertical size of the beam? And so, for example, part of this project is looking at plant roots. This is a corn plant, and we're going to be imaging the roots down here at the bottom. So these are the plant's been removed, the roots washed off, and this part of the plant here. So this we imaged at the light source in soil, and so with that imaging technology, we can actually see the roots and see them quite nicely. What we're relying on here is something we call phase or inline phase contrast. So preservation of what we call the phase of the beam is essential in order to have this. And because the beam was small vertically, there was about 4 slices that were taken, or 4 spins of this object, and they were stitched together to create the image that you see. So it had to be done 4 times in order to do that. And so it would be very handy if we could do that all at once, especially in a system that might move on us. It takes time to do the stitching. And so it'd be very handy if we could expand the beam and preserve the phase aspects of the beam. So we used a system that involves X-ray diffraction. There are crystals here. There's the first crystal here that gets hit by the raw synchrotron beam. This crystal is highly bent. It expands the beam very rapidly. That expanded beam hits the second crystal where it's thrown horizontal, and then we use this as the imaging beam. So if we do this right, we can preserve the phase of the beam and now have a vertically expanded beam, and we haven't touched the horizontal aspects. So this is what it looks like. This is the first crystal. You can see the crystal has been on a frame. This is the crystal, this shiny bit here. This is the second crystal looking at the backside. There's a big slot here to let this beam enter that crystal and then be diffract horizontal. And this is looking along the beam path. This is the first crystal. The beam goes up to here, the second crystal, and then is thrown horizontal by that crystal. So this is what it's done in one of the first experiments that we did. This is a mouse in a centrifuge tube. You can see all of these little lines here. This is tape that we use to hold that object, and you can see quite clearly the edges of the tape. In a radiograph, you would never usually see that kind of structure. This is what we mean by the phase contrast aspect of the imaging. This is a test object that has edges on it, and you can see the bright edges here. This is, again, the phase contrast. One thing to note here, this was the original size of the beam, which is about 5 millimeters. In this experiment, we expanded the beam, in fact, over 50 millimeters, vertically left the horizontal alone, and you can see how many images we'd have to take, over 10 images in order to cover this object with the beam size we would normally have. Just blow up a region here just to highlight the fact that there is a phase preservation aspect, and that's this bright line you see around the object. And again, you can see this crinkly stuff from a foam that we use to create some phase contrast. There's an optimization process that we've gone through for this project and to try to optimize it for this imaging technique for the plant imaging. There are a number of things that have to be optimized. I won't go into it. There's a program that's been used in order to do that optimization that helps us in the optimization process. This is done by Dr. Xian Boshi at the Advanced Photon Source and a former student of mine, Dr. Nazanin Samadi, who's now at the Swiss Light Source, where they're also interested in this technology for their imaging program. I won't bore you with these numbers, but this is some of the calculations that are done, imaging energy and other parameters that are found or either optimized for this system, for this application. In order to assess the phase preservation, we have a test object that puts the beam on the ledge, and what we're looking for is for the edge of these beams and how much that edge has been affected. This is detected somewhat downstream, and you can see the pattern of those beams and a bunch of numbers that assess what we call the phase preservation of that system. So far we discovered that when we optimize the throughput or the intensity, we degrade the phase, and so we've gone into a study trying to understand that. There's a theoretical paper by one of my students, Pian He, who's working on that. We've done a number of tests to try to assess the phase preservation and the utility of this system, a number of parameters here to trying to optimize that. This involves crystals made at the Advanced Photon Source for us and used quite a bit of beam time in that assessment, 24 shifts, 24 8-hour shifts, and that's over $100,000 in equivalent cost as part of my beam team allotment. So next steps, we're going to go back and implement the original system because it preserves the phase quite well. forge ahead and try to design a better bender and fabricate that. Certainly the theory, that's very critical for understanding how the systems work and understand the phase preservation aspects. Measuring the crystal shape is critical, and of course, test that system. So with that, I'll stop and thank you for your attention and can entertain questions. Thank you.
Hi, everyone. It's a pleasure to be here participating virtually in this year's PERC Symposium. I miss you all. I've really enjoyed our time together in previous years at previous events, but I'm also happy to be here at the University of Ottawa sharing some insights from our Ottawa-based team on the topic of intellectual property, ownership and control, and international trade law in relation to data stewardship. Data stewardship is a topic that causes a lot of confusion, a lot of uncertainty, and can be very intimidating. It can lead researchers to stall projects, uncertain about whether data should be kept secret or shared or sold. And so one of the things that we're working on are open data initiatives like the FAIR principles, the principles that data should be findable, accessible, interoperable, and reusable. FAIR is just one example of some of the latest open data initiatives. These are becoming increasingly popular. And I think many of you have questions about what these mean for your projects. If you have those kind of questions, reach out and get in touch. We'll be happy to discuss. What we're working on as members of the social scientific research team are what your projects and your use of data actually mean for the broader ag biotech community, and in fact, the scientific community at large. So we're looking at these open data initiatives to try to understand how they operate, whether they're effective, in what situations or circumstances they're effective, and when they might need to be nuanced or alternative approaches adopted. What these open data initiatives typically have in common are a set of principles that all researchers, funders, industry partners agree to upfront. And upon accepting those principles, there's some kind of branding or certification that takes place where the findability, accessibility, interoperability, and reusability of the data exists by virtue of this kind of certification process, because everybody gets on the same page about the common objectives. We're also doing a lot of work with international organizations, UN agencies like the World Intellectual Property Organization, or most notably the Food and Agriculture Organization of the United Nations. And with these international organizations, we're looking at the frameworks that will govern the issue of data ownership and accessibility at the international level. So countries right now, and we're working very closely with the government of Canada, as well as developing country governments, to input into these processes happening at the international level, which will lead to a legal framework governing control over data. And that's going to have an impact on all of our research, maybe not tomorrow, but certainly in the medium and long term. The other thing that we're doing is we're looking at hotspots around international trade law and international trade agreements, and how this will impact your use of data, whether it should be kept secret or shared or sold. And what's happening in these trade agreements is actually sort of a counter movement to what's happening at the grassroots level with researchers, scientists, industry partners, and funders. And in these trade agreements, we're actually seeing a ratcheting up of legal protection for trade secrecy, creating the legal conditions to facilitate secret information. So contrary to making data findable, in fact, the legal framework is being changed to make it easier to keep data secret, because it's often incredibly valuable in various sectors. The other thing that's happening is there's a real push to protect agricultural chemical test data at the international level. Now, in the context of COVID-19, we've probably heard about the importance of clinical trial data for vaccine testing. So we need to know whether vaccines are safe and whether they work, whether they're effective. Well, the same thing happens with agricultural chemicals. Before chemicals are allowed to be sold or marketed, and this includes fertilizers and pesticides and herbicides, before this is allowed to be sold and marketed, they need to be proven safe and effective in terms of human health and environmental safety. Well, the data that's being used, and that could be microbiome data, it could be genomic data, proteomic data, or phenotypic data, the data that's being collected in these field trials is actually protected against other people's use. And so that's one of the big issues happening in the international trade arena. So the social sciences team at PERC is complementing what's going on in all of the flagships by studying the international trade law framework, by studying what's happening at international organizations, and by studying the practical implementation of open data initiatives, and learning not only what these initiatives mean for you and for us and our project, but also what our project and our practices mean for the broader movement to promote open data and the legal framework that enables that. So that was just a quick overview of what we're doing. I can't wait to see everyone again in person at the next symposium. Until then, take care and look forward to talking virtually. Thank you.
All right, terrific. Welcome back, everybody. Thanks so much to our presenters for those excellent presentations. We've got Drs. Wagner, Mamet, Stanley, Chapman, and De Beer here with us in our panel session. So we have had a few questions filtering in through the chat, which is terrific. Hopefully at some point during this panel session, my brain doesn't overload at all the different platforms we're interacting with here, but I'll do my best. So I think what we'll do is start with a couple of questions that came in through the chat after Dr. Wagner's presentation, and we'll just sort of step through things that way. So the first question that we had was a question about whether or not there were any pesticides applied to the plants for the microbiome work, and if so, how that might affect the microbiome composition in the lines or groups of genotypes.
There was just standard agronomic conditions we used on our research farm prior to this experiment. So there was a pre-emergent herbicide and some early applied insecticide, and I don't remember off the top of my head what those were. That's an open question about what effects they might have on the microbiome. And I don't know enough about pesticides, honestly, to speculate about how that might interact with the plant genotype.
Great. Yeah, I know that's a question that we often grapple with in my area of research as well. I mean, do you treat your field crops like farmers treat them when you're doing microbiome research, or do you create artificial conditions in order to avoid any potential impacts of pesticides? So it's a tricky one. Next question about your work. How many sampling time points were there in your first study, and do you think that you would see those effects throughout the lifespan of the corn, or how does that work, do you think?
Yeah, that's a really good question. So to answer the question, we only sampled both leaves and rhizosphere at a single time point. For leaves, that was about 3 weeks, and for the rhizosphere, that was around 7 weeks just before male flowering. The reason we chose that later time point for the rhizosphere community is because we thought that throughout the plant's life cycle, the roots are acting more as a carbon sink, basically until it transitions to the reproductive stages. And so any impact of the host genotype on the deposition of carbon into the rhizosphere would be sort of building up through the plant's lifespan up around until it starts to reproduce. So I wouldn't be surprised if after the transition to reproduction, if the effects of host genotype were lessened. I also think if we had sampled earlier, we still would have seen effects. For corn, heterosis is obvious basically as soon as the seeds begin to germinate. And so we know that there's something going on with the hybrids as opposed to the inbreds right off the bat. But it would take kind of a more finely tuned time series to really characterize the dynamics of where those results show up. But I would expect for the differences between hybrids and inbreds to sort of build up right until around the end of the vegetative stage and then kind of decline again.
Great. Yeah, I'm just wondering if our other panel member, Steve Mamet, might want to comment on that, because in the PERC canola work, we actually had a fairly intensive time series and looked at some of those patterns. Steve? You're on mute if you're speaking, Steve. I'm not sure who's got you there. I can't. There we go.
Sorry, could you repeat the question, Bobbi?
Oh, absolutely. So Dr. Wagner was just we were talking about time frames, and her comment was that if indeed they had sampled later, she expects they might have seen a lessened effect of plant genotype, or if they had sampled earlier, because heterosis does set in very early in maize, they still would have seen those impacts, but it would take a more highly resolved time series to really interrogate that. And so I guess I'm just wondering if you can comment on our pretty highly resolved time series in the PERC canola work, and what we observed for the most influential times on the microbiome there.
Yeah, we didn't explicitly look at a time series of the genotype effects. We did a bird's eye view of that, but some of the more influential times are around flowering, if I recall. We've done a lot of passes over these data, and I'm just trying to think back to the qPCR when we were looking at when are the effects most pronounced over the time series, and I believe it was about mid, which was around flowering for us, and it seemed to very low at the start, and it seemed to taper off at the end as well.
Yeah, that's my recollection as well, as the tightest linkage really between the root microbiome and genotype effects were at flowering. And so I think we honed in on weeks 6, 9, and 12 after planting as sort of key dates, which were 3, 6, and 9 in our experimental design. Great. So another question for Dr. Wagner. Do you think that there will be an impact of these, or what do you believe the impact of these results will be practically for plant breeding?
I think we're a really long way still from being able to sort of use this type of data directly in breeding efforts. The heritability of, for example, like OTU abundance or something is just so low that I don't think it's worth the effort to even try to do that. Not to mention that we don't have a good understanding of how microbiome composition measured through amplicon sequencing maps onto function in any reliable way. So I think there's still quite a lot of work to be done. However, I do think most of the studies that are being done are finding some effect of host genotype, and that's on this amplicon-based data, which, as we all know, has a lot of limitations. It's a simplification. There's a lot of evidence coming out that's less important than the taxonomic composition of the community might be the functional community or the functional profiles and activity of the microbes, which to my knowledge, there's not very much data at all on how heritable those properties are. And so if there's a lot of functional redundancy within the community, I think it's not out of the question that someday these factors could be sort of considered in breeding efforts. But I do think that there's really a lot of work to be done before that's possible.
Great, thanks. One last question in the chat here regarding your presentation was, do you think these effects are, because these effects are plant driven, that they would be transferable between different site years, or would specific environmental factors override them?
Yeah, that's another very good question. In most studies of plant microbiome composition, the location or environment has been the dominant sort of explanatory factor, almost always sort of swamping out the effect of host genotype. Again, and that's when we're measuring microbiomes using this amplicon sequencing approach. So you've got its taxonomic composition. So we're, that environment effect encompasses both just microbial biogeography, so which microbes live where. Just by that fact alone, you're going to have a strong environmental effect. But there's also phenotypic plasticity involved, where the environment's interacting with the host genotype to influence the habitat that these microbes encounter. So in the study that I just talked about, those results were averaged over 2 fields. And so if you dig into the genotype by environment interaction, you see stronger effects of host genotype as expected, and that's in line with what most other studies of this type have seen. So I guess that was a long way of saying I do think environmental factors are probably going to override the host genotype effect, especially as you start including, a larger diversity of environments that are just starting out with different ambient communities.
Terrific, thanks. There were some sort of related questions for Dr. Stanley. I'm just trying to sort, they're a bit not in order here in the chat, but I guess does the problem of creating interconnectivity in these complex microbial networks become easier as more genomes of soil bacteria are sequenced, or do we really even have a good idea of what bacteria exist in soil? Kevin?
Well, I can't answer the second part, but my impression is no. I'll leave that to the soil microbiologist. But so tricky question. The short answer is no, because n3 to the n is still n3 to the n. There are some benefits, though. This particularly comes down to the idea of ASVs. If you could start collapsing ASVs into like logical genotypes and be like, no, this thing is actually not that different, we're going to count them all together, right? You would get rid of a lot of those columns. That would increase the density, would make fewer zeros. That would make the data cleaner. Cleaner data leads to fewer squirrely things when you start doing pairwise differences. Fewer squirrely things means better conclusions. So it would be better, but it's not going to solve the core issue.
Okay, great. Yeah, and I mean, certainly technology-wise, it has been an improvement to be able to move to this amplicon sequence variant, whereby things that differ in their DNA sequence by one base pair can be resolved as different things, but are they functionally different? And if we want to regroup them into simpler groups more related to their function, how do we do that? And that speaks back to what Dr. Wagner was talking about a minute ago, and the idea of really phylogeny or DNA sequence-based identity, not necessarily reflecting the important functions of different microbes. So.
Yeah, and I think that's actually part of the motivation for the whole building these ecological graphs, because if you've got things that are very close together genetically and they're doing exactly the same things with all their neighbors, then maybe we should just treat them the same.
Terrific, thank you. So I guess a question that came in after Dr. Chapman's presentation, what are the prospects for developing high throughput, easy to monitor, i.e. digital imaging above ground proxy phenotypes that represent well-characterized below ground phenotypes, so root microbiome structure? This came in after Dr. Chapman's presentation. I'm not sure if anyone on the panel wants to volunteer to field that one, but can we measure proxies above ground for what's going on below ground in a meaningful way to make all this below ground stuff that we know is so important a little bit more accessible? Have anyone that might want to tackle that?
I can talk about that a little bit, but I'll let Dean go first.
Okay.
Pardon?
Do you want to take a run at that?
I don't know that I can answer that. I'm a physicist by training, so this one will be hard on me. I mean, I think the interest was to see if we could see what was below ground is the primary motivation of what we were doing. Above, there are many technologies we can use, including X-rays. We use X-rays to see below ground. So we can see structures, microtomography, we can see very small structures of things above ground. But I think this was all motivated with trying to do high throughput beneath the soil imaging of roots. That was the whole motivation. And I can't go much farther than that. You want to talk about crystals, I'm in, but
I guess, Kevin, did you have something that you wanted to comment on?
Yeah, so this was kind of an idea that Steve and I have been throwing around for a while that seemed ludicrous and then didn't seem so ludicrous and maybe seemed ludicrous again, And this was the idea. Could you tell what kind of bacteria were in the soil from a satellite image? That was the question. So if I took a picture from space, could I tell you what kind of microbes were in the soil? And the short answer is no, of course not. That's crazy. But then you think, well, maybe not, but maybe if we had these functional groups like we were alluding to, we might not be able to tell you which exact ASVs or species are there, but there's got to be things of this general genus doing this kind of stuff if the plant is behaving that way. And if we have a big enough data set, then you could conceivably throw some machine learning algorithms at it. I think Felix asked a question that was kind of related to this. Well, yes, but that's great, but doesn't that assume you know what all the microbes are already? And that's true. In this case, it would be more about which microbes do we think are there from the microbes we know should be there. So if you trained your magical system that we've envisioned that maybe isn't crazy and maybe is, on canola in Saskatchewan, and then you looked at a rubber plantation in Indonesia, it would give you ridiculous answers. So you'd have to, it would be more of a monitoring rather than surveying kind of functionality.
Okay, great. A question for Dean, something that I've always wondered about. I mean, you were talking about phase contrast in your presentation, and this morning we saw some work on abiotic stress, like drought stress, for example. Could you just speak maybe to some of the challenges of imaging roots under different water conditions, like the interference that water, obviously so important for plant growth in soil, has in terms of trying to image roots?
Yeah, I mean, the problem and maybe the opportunity with X-rays are they go through things, but there's very little difference between the tissues of the roots and the and water itself. So what the X-rays see is a very subtle density difference between. So really, what's showing up are the what's outside the root, the skin of the root. I probably should know more what you would call that. But that very subtle density difference is what causes the phase contrast and allows us in imaging what you call to segment them out so you can see them separated from the water that surrounds them or the soil that surrounds it. So density is everything in this business. A little bit about the compositional piece, but primarily in this system, it's subtle density differences that show up. Is that helpful? And I should probably know what you call the skin of a root, but I don't know what you call it.
That's all right. Okay, terrific. Thank you. A question for Dr. DeBeer. If an organization believes their private data is valuable, what are the mechanisms for that organization to make their data open and fair, but also extract value from it themselves?
I'm sorry. There's just a little bit of chaos going on in the house right now. It's time for the girls to get ready for dance. They're off to rehearsal. So I missed the last part of the question.
You bet. Let me try again. So if an organization believes their private data is valuable, what are the mechanisms for that organization to make those data open and fair, but also extract value from it themselves?
Well, I think it depends. The question is, what kind of value? Where's the value coming from? And is the value in terms of a scientific breakthrough? Because if that's the case, then making the data findable and accessible and interoperable and reusable could be the best way to realize that value. Data from Saskatchewan and Indonesia need to be interoperable, and maybe some of these problems that Dr. Stanley was talking about can be solved not by one person but by a team of collaborators. If the value is professional, then open data can actually be a wonderful way to realize professional benefits by, for example, increasing citation counts to your publications. One of the most powerful multipliers is when you have a data set that is used and cited by other people. So if the value is your H-index, well then open data is a great way to go. But if we're talking about commercial value, then that might be something different. And in the context of commercial value, you're going to have to work with your industry liaison or tech transfer office, or your business partners or your investors, or your funders, your government funders. and try to come up with some sort of a strategy as to how you're going to pursue that kind of commercial value. And there are a variety of different models. Secrecy and exclusivity, closed data are not necessarily the best commercial strategy. Often open data can lead to really valuable partnerships where it's the peripheral knowledge and the team around the data that's really of interest to the commercial partners. And what they're paying you for is not the data per se, but the knowledge about what it means. And just sort of to wrap up that last thought, I think you've really got to understand what kind of value it is that you're interested in extracting.
Yeah, an additional comment was specifically with respect to monetary value, I guess.
Yeah. Okay. So then again, in that case, if it's the monetary value, I guess one of the myths that we need to overcome in the scientific community, is the likelihood that our data will be commercially valuable. So very often we have in mind this lottery ticket ideology, which is not a very realistic view of the true commercial value of the data. When you look at the proportion of projects that actually generate blockbuster returns, it's infinitesimal. And so what we've done is too often we've set up a system where in pursuit of the lotto ticket, we actually generate all kinds of transaction costs and inhibit collaboration and more modest realization of value, thinking that we're holding a winning ticket when in reality that's exceptionally rare. So I think that what our research is showing is how important it is to be realistic about the likelihood that the data that you're collecting or holding is really as valuable as you think it might be.
Right. Yeah, I know at the end of your presentation, when you were talking about sort of that protection of agrochemical data, it made me think about the big bit of momentum that we're having right now with biologicals and the idea of protecting that information and how it impacts researchers' willingness and enthusiasm for engagement with industry. I mean, if you feel like you might have, like winner chicken dinner, I found the magic microbe. Does that make you feel less likely to make those engagements or more? I don't know what the typical pattern of behavior is, but I could certainly agree with, the odds are long.
I think it's really context specific. It so much depends on the particular technology that you're talking about, the particular application, the particular industry partners. And so the question that I think the scientific community needs to ask itself is, what kind of error do we want? Do we want to err on the side of being overcautious and overly secretive and thus inhibit the academic rewards and the scientific breakthroughs, holding out hope for a commercial success? Or is it worse to err on the side of letting loose and maybe, just maybe, you've given up a losing lottery ticket or a potentially winning lottery ticket, but in return, you've realized far greater benefits of a different sort.
Great, thank you. I think I've gotten to the end of the questions that we've had coming in through the chat. I'm just going to wait and get confirmation on that. So that is right. Are there last minute questions from the audience that you have for the panel this afternoon? All right, well, I don't see anything coming in. So thank you very much again to our presenters this afternoon. It was a really informative and interesting conversation that we've had. I guess I would just like to remind our audience once again to visit the poster gallery. There's lots of good P2IRC research that's been showcased there. And then just remind everyone that we're back again tomorrow morning at 9. And we look forward to seeing you all again then. So again, thank you very much to our presenters and have a great afternoon, everyone.